Swayam Prabha. Digital India. Educated India.
Hello all, welcome back to the course on Precision Oncology. This class we will be learning about role of drug discovery in precision oncology. Last time we have gone into the introduction of precision oncology, how exactly it has taken its shape today. We studied about few models about field character cancerization or cancerization of the field. We got introduced to different terms. Why exactly precision oncology is very important for cancer biology. We have studied about certain cases about your squamous skin carcinoma, and then we went on to study about the companion diagnostics, which are playing a very key role in cancer biology. And then we have really known what exactly, how can you be precise for cancer diagnostics, and how can you be precise with your cancer treatment. So we really understood, if you recollect, we understood with that stick diagram that how precision oncology should be both precise and accurate for giving a very good deliverable in terms of a better targeted therapy or a better diagnostic marker. Now, moving forward, we haven't explored the role of what are the current drugs, what are the exact current clinical trials, and you have to process of what is called drug discovery. What exactly is drug discovery? How does this process? How is it? Is a very much why do you need the drug discovery process? Now, first drug discovery started from ancient methods, like for example, you were having curcumin, or you have even your neem extract, many as in the as drugs now, but or your papaya extracts, which were reported to lower the cell counts, WBC counting during leukemia. So now, how do you really go about identifying a compound? It's not only naturally occurring or medicinal compounds, but even your synthetic compounds. How exactly do you go? There is something called what is called your drug development process, where you have your target identification. For example, where exactly this particular drug, maybe not necessarily cancer, even in your regular antibacterial or your antibacterial. What exactly, where exactly, for example, your cell division protein or your cell wall, like your beta lactams. You will identify your target identification. Then you go for your what is called your lead discovery. Okay, then you will look for the different compounds which could be targeting this particular, or your beta, for example, your beta lactam analogs, or your isoniazid for your MTB analogs, or any other different. Then go for your medicinal chemistry or maybe one more approaches. Use your medicinal approach, and sometimes even in your in silico studies. This is your dry lab, where you have many drug libraries, and then you look for analogs which are similar to your existing drugs, and then you further. What is the whole idea of this drug development is like to find better novel drugs all the time. There is always, because of your resistance studies. Then after you're successful, then you will look for all. First thing you will finish your in silico, and then your in vitro studies. What is the in vitro studies? To your simple antimicrobial testing or anti-cancer. You take many cancer cell lines. Supposing you want to come up with your breast cancer drug or triple negative breast cancer, you came up with a very novel compound, something better, like tamoxifen. It is very much toxic. You come up with a compound which has better permeability. You say that it will have a better IC50 or better validation. You can be with this particular. You select your breast cancer cell lines and do so many in vitro studies, like your MTT assays, like your apoptosis assays, then many assays to show that this particular drugs have anti-cancer properties. And then after my all this is done, I have shown in vitro, I have shown in vivo, then I will go for in vivo studies. So first we take the nude mice models, where you put all the cells, or maybe for example you are taking only the cells of this particular your your breast cancer cell lines, like say for example 231. You put those cells, you induce tumor formation, and then you give your inhibitor. Then further go on with, then this has sustained the mice. Mice have responded well. There is no, this drug particularly has able to contain your tumor. Then you go to your clinical trials. You have your, there is much of your, and again you have lots of in vivo imaging solutions to support drug development. That is like, you have where exactly is this drug able to, can you track it? Yes. And then you have your, this is the path workflow. For that you have the model development, then you have the target validation, then you have the, you measure your different assays, and then so forth. When it comes to clinical trials, this is very simple basic diagram. It may not be as simple as this. You have the preclinical trials, which are like your phase 1, phase 2, phase 3. It's just not necessarily with respect to the participant number. If it is like, if all your preclinical criteria are taken up, it goes for the phase 1 trials, where a small number of participants are taken up. Then to the phase 2, where you have, you know what exactly, yes, this particular group of people have responded to the novel drug. Yes, I will go for. And first thing, very very toxicity. First thing, it can't be just like that you have a drug and all that. And moreover, you have to see the solvent wherever you are putting up in the drug. You should, maybe your drug is very solid, very insoluble, you put it in a very strong solvent, and that you can't bring it up to this level. These are all very very important factors uh, for your drug discovery process. Then you have around, as you have increased for the phase 3, phase 3 here, where you have 100 to 3000 participants, and then it will go for to safety and effectiveness of the drug. And then you go for the phase trials, phase 4, where mostly after the FDA reviews, after the FDA gets it, the draft gets approved, and it is maybe mostly in the market, but only the last phase will only include the final of your safety and effectiveness. First thing, for a drug or for any to try it up a novel thing, what was the earlier approach? It had mostly of your histological classification. In earlier, oncology patients were generally classified by the primary cancer, which we already said in your cancer detection methods, and then your stage of your cancer, TNM methods. And to have your control trials, your randomized control trials, you need to be, you had to have, each patient population would be going through the same standard therapies. Based on this particular perspective, all the existing cancer drugs were developed. Now, as we have seen so much of work coming upon the mechanisms of cancer biology and all that, so we have seen how the cancer cell growth and progression has been defined at molecular and cellular levels. And so we have seen there are so many markers, molecular markers at the genetic levels. You now don't classify only according to the histology, but you even give a morphology class subtype to these tumors. It is called molecular profiling or molecular stratification. So this is a new term which we are coming across. That is why it is no longer only this particular broad cytotoxic agents, but there we are also employing your molecular targeted agents, which they will act perhaps only on selectively on cancer cells. For example, your TKI inhibitors, your tyrosine kinase inhibitors, which are very well used in your lung cancer. This molecular targets, agents that target specific molecular markers include your gefitinib, erlotinib for the EGFR gene mutation positive, for your recurrent metastatic non-squamous lung cell carcinoma. For your ALK fusion positive, we use the crizotinib, alectinib, and ceritinib. We are also using, there is a lot of change in the whole scenario of employing targeted precision therapy for that particular cancer. So how did, so you had two types of trials. So ones were patient centric and some were drug centric. So traditionally they were more of drug centric. You identify the function of the drug and then you administer it to the patients. So then but how do you when you go for a novel drug, maybe as I mentioned in the phase or for your clinical trials, how do we go when there is when you want to aim to identify certain attributes of the patient? So you have to be very specific. For example, your genomic aberrations, for example, this SNP polymorphism or this particular mutation. So can you put this patients in this particular drug regimen? There is a large variability in your genomic subgroups, your microenvironment, your baseline characteristics, and all this, your your patient population who is taking a particular drug, there are absolutely a heterogeneous population, which is not only specific histologically, but also your gene agnostic trials. This is very, your phase 3 randomized trials are very critical for regulatory approvals. Since the anti-tumor after was, you have two arms. You have the comparative arm, and usually you compare, you have the conventional therapy as a comparative mark, and then maybe my therapy, whatever, like if I come up with a new drug, then I should have a randomized control trial. So how do I go about this usually, and what is the end point? How do I say that this particular drug is effective? You even have so many other parameters to judge that. This is like our very favorite line for this whole course is one size fit all medicine is not there. It's not now, no longer this one. For example, just imagine you have the therapy which is fitting for all patients. Some patients take the adverse benefit, some patients will benefit, and some patients will not have benefit. This is how the general traditional. Now from here, patients are grouped by your subtypes. You are stratified. This whole population you have stratified as based on your risk profiles. Then you say on your demographic, for example, your Caucasians group for lung cancer, the mutation profile, how does it differ from the Europeans? And then socio-economic of their, maybe for example, if they are working in very coal factories or any other chemical factories, which we have seen molecular mechanisms of carcinogens, if you recollect. And then clinical features. This is also very, very important. Some patients may come up with symptoms, some may not present themselves with some symptoms. Then biomarkers. You are different. Your companion diagnostics are very, very important here. You are creating a molecular subpopulation. You are coming to something what is called precision medicine. Individual patient levels, how is their molecular tumor different? Their lifestyle, their preference, their health history, any hereditary cancers, complaints, and then exogenous factors. All this which we have discussed. Now, when you are giving your drug treatment, can you see here? You have the three different. You have a different kind. Here, see, it is a same drug here. But whereas those two colors, because you are able to stratify, for example, patient with one mutation, this mutation is wild type in one patient, for example, and it is a mutant. You are able to, supposing, as I told you in our earlier class, you don't want to unnecessarily burden the patient with drugs also. You know that he is definitely a mutator. Suppose he is a wild type and you may not have a specific drug for that wild type tumor. What we need to, you don't want to burden with the patient with the adverse effects of the drug. You are coming up with something what is called your individualized therapy or individualized therapy. There is a treatment shift in your treatment strategies. That is as a result why we are totally employing a precision oncology drug. Now, what all do you patient centric trials? So there are as I told you, they were drug centric. You should avoid usually unvalidated surrogate endpoints in the recent drugs. You should have it should not have more just side effects. You cannot be using definitely any drug, no generic drugs, and they shouldn't be biases in clinical design, maybe like favoring one particular compound over another due to any other interest. And overly restrictive exclusive criteria should not be there, should not be adapted. You should, this particular patient centric trials, they should have hard endpoints or validated the surrogates. You need to have a pakka endpoint, which is a very, this endpoint could be like mostly you measure with your overall survival, progression free survival in the cancer patient. You have linear trials with higher intended effective sites. Then you should be able to promote this particular patient centered trials, should be able to be show a promise for investment in R&D and biomarker discovery. So these particular trials are fundamental for achieving better outcomes for the precision oncology. This is not very perfect trial, may not be able to achieve, but we have to ensure that this particular any trial has to be their best. So and every patient has to be definitely be informed about this trial, and they have to be ethically approved as to see to it that we are in given guidelines with the international boards. We even have we have mentioned very briefly last time. So if you recollect, you have the molecular monitoring of patients treated with precision therapeutics to demonstrate your proof of mechanism. For example, you really want to show how your action of the drug is there. You give, for example, we have a breast cancer or a lung cancer patient, and you really want to give a novel therapeutic, say for example, say an FDA approval drug to the patient. I want to try it a new one. But before you give it to the patient, you should be going through your non-clinical model, your mice models. So that you excise the tumor from the patient, and it is put onto the mice. And from that you will go for after it goes with onto the mice, you will give it the same inhibitor. For example, your gefitinib, what it gives to the patient, you give it into the mice. You will really see, is the tumor able, is the mice able to, oh, still is the tumor surviving in the particular mice, or is it like in the presence of an inhibitor. Sometimes the tumors overgrow the inhibitor. It's not that the mice will be healthy, but still you measure your size of the tumor. In that case, there is every likely event that the patient also may not be responsive to that particular drug. Here itself, you are having another model where you can particularly even see for a patient, for example, you may require 2 to 3 months to monitor the effectiveness of a drug. Whereas for a mice, another 15 days to 20 days, yes, we can know. You can slowly, we can really predict the models, the efficacy of the drug using this particular mice model. And then you go for your other, like you go for the sequencing for the same tumor before you implant it into the mice. Then you look for the methylation, then you for the fish, then the IHC, and then you look for the expression area. For usually patients are eligible for early or late phase clinically trials. We analyze the tumor and other tissues for pathway activation of resistance, and then based on the molecular characterization of the tumor, for example, if it's a mutation positive here or a wild type, the patient is assigned to that particular trial, and then the patient is particularly monitored, and then post treatment also we go for several molecular reanalysis, and then different clinical observations. For example, your PET CT, then you have the functional images for your now the prognostic for your normal and tumor tissue, predictive diagnostic markers, and then your and then you go use your CTCs and your CECs. This is like you have the molecular mechanisms to really demonstrate your proof of mechanism for proof out for the development of targeted therapeutic agents which focus on which have focus on the use of your preclinical models to validate biomarkers that can substantially be with tissues obtained from patients. So this is a very important workflow algorithm for therapeutic molecular monitoring of the novel therapeutics developed. So this is exactly what I was trying to tell. You have your drug discovery and your drug development. You have the target identification and validation, and then the lead identification. You identify the lead compounds, and then preclinicals. Then you have the phase 1 and 2 and 3, and then you look for the exploratory mechanistic pathways. Now, what exactly are your readouts? What do I mean by readouts? How do I really measure? You need to have a measurable. For example, you do an ELISA, you have a spectrum reading, or you do any other calorimetric assay, you have a reading. For how do I know if my drug has worked on any of my models? This is like I use different models. Now, first thing here, 2D culture. For every cancer biology, we use MTTSA. That is something in the 2D culture where you just coat the cell lines onto a plate that you have the cells growing, and then you had your inhibitor where you assume that the cells numbers have decreased and you just look for a simple viability assay. This is how your readouts are, it's cells viable. If your inhibitor, you have the viable cells here and this is the dead cells. If your inhibitor works, your cells would die when compared with your control where you, it's very important. You have a chance that it is very low cost. You can even have 90 from 96 well plate to a 384 well plates, and then it is many more drugs, and it's not only one two, you can do it for many drugs. And then, but it is like it is not, it is a bit dependent, definitely no more longer. If you stop it at 2D, nobody will accept it. You cannot, and moreover, if it, you won't have a tumor microenvironment, which is very well missing over here, because it's only a pure homogeneous cancer cells, whereas it doesn't happen like that, right? When we have seen tumor microenvironment where you have your cancer associated fibroblast, and we have other stroma cells and other immune cells as part of it, all that will be missing, which would really influence the growth of your cancer cells, right? And definitely these culture conditions, whatever we grow is 5% CO2. It's not very, it's an artificial culture. Something a little better than this is your 3D organites, where you just, as I mentioned, dissect your surgically obtained tumor tissues, and then you put it in a gelatin scaffold or on your matrix gel or whatever, that, and allow it to grow in an artificial medium and temperature. Yes, the cells grow, the tumor cells grow, and then along with them you even have, for example, for your pancreatic tract adenocarcinoma, you have the stellate cells growing on them. Yes, you still say, yes, my 3D environment, I'm able to bring the tumor microenvironment. It's not very costly. Yes, it can be scalable for high throughput, and then it is amendable to rapid testing, assay development, and all. However, a prolonged, you cannot grow this, maintain for maybe not for several. Here, supposing you have 2D culture, you have thousands of generations you can maintain, but here no, because it's by virtue of its own. Until unless you extract your stellate cells or any of this tumor cells and then you immortalize them, you cannot do that. That sometimes, yes, because of you can't take more than a small few for your organoids. You end up missing some of the tumor microenvironment and non-physiologic culture conditions. Then you have organ on chips, which is very, very widely used nowadays. Then yes, it can be going for your high throughput sometimes, but not always. And the patient-derived xenografts, where you have your mice model where the tumor is put into the mice, and then you can see how the tumor is formed here. Can you see that? You still maintain in vivo tumor microenvironment. You can show the drug pharmacokinetics. Yes, can it be toxic? Yes, we can have the parameter. But however, it is very costly, and it is like tumor host species. See, for example, from human to mouse, there is a host species difference, right? And then, of course, these are nude mice. You don't have a immune system. What will happen if you have any of this, not a nude mice and an ordinary mice? The immune system will definitely recognize your cancer cells as foreign, and definitely they could, in all probability, they could be eliminated. Yes, this is a very, very important part in your human patient also, where you have, yes, in the trials. Definitely when some patients now, many people are enrolling for any rare tumors and all. Yes, this they are, of course, they should, all these particular drugs, whatever novel drugs we want to test on the for this novel patients, they have to pass through all this particular criteria, and then only the patient, and more importantly, the safety and efficacy and non-toxicity. You don't have to have a model establishment and all that. Yes, very, very important part is there's a high-end regulatory barriers, not very well permitted, and it's very important to have a lot of regulations, as I mentioned, because of the human samples. And they are have to be accessed. Everywhere you have the gene expressions, then you look for the tumor volumes. For example, here at this particular organites, you look for the size of the tumors, then you look for cell mass and morphology, and then If it's like a micro device, implantable micro device. You have now role of how, what is the role of your pharmacodynamics and diagnostics in precision oncology. This is very very important for your clinical trials or to progress. So how do you have your clinical trial designs? In this coming slides we will really see what exactly is your clinical design. How do you group a clinical design into your I mean, for example, are they patient specific, or how do we have the drug specific? Now it's like it's based, your clinical design, trial designs basically on because of the are based on molecular markers, and they have gained because of the changes in the chemotherapy agents. Yes, they have got a lot of popularity. So trial designs called enrichment designs or targeted designs, where patient populations with a single molecule for a molecular marker for which a drug can be expected in a specific tumor type. In one single tumor type, yes, is there a single molecular marker for which is there any changes in this particular marker expression? This design is selected on this particular premise that the molecular marker has to be an established marker. For example, your HER2. It is supposed to be which is strongly correlated with the efficacy of an investigational drug. It has to be biologically demonstrated that drug test efficacy cannot be expected in marker negative cases. For example, if I don't have a mutation, this drug will not be working. And it is a diagnostic for, and can have you developed a diagnostic tool, for example a qPCR, or for or an NGS, or a simple PCR also for can you measure this molecular marker, and not necessary, and this molecular marker shouldn't be measured only from a biopsy, but as the treatment progresses, you should be able to measure this particular marker. And how can we do that? As I mentioned before, using your cell-free DNA, your ctDNA. You have a drug, you have the molecular marker, and then you have the functional consequences. To evaluate your therapeutic regimens for your cancer based on the degree of target engagement and the downstream molecular initiated drug target interaction, you need to have what is called pharmacodynamic markers, PD markers. Very important. They are molecular species that are altered in response to an oncologic drug or treatment, and if measured can be used to establish your action mechanism of action of the drug. PD marker helps to define the drug induced pharmacological effect associated with the therapeutic activity of the agent. A PD effect does not by itself ensure patient benefit, but benefit because biochemical events downstream of the drug target may impair tumor cell care. We just can't say that I have a drug, I have a marker, chalo, I have totally gained, I have mastered the tumor. No, it is, as I said, it's a very intricate mechanism of, very beautiful mechanism of cascade pathways. It may be like, maybe what if the one point of action the drug is able to inhibit, but further downstream of the drug target, it may not be very active. Your pharmacodynamic biomarkers may not be predictive of patient benefit from a specific intervention. This is development of a predictive biomarker is the goal for precision oncology. For the first step in the process is to establish your molecular proof of mechanism in molecular proof of mechanism POM in human tumors, and you have to establish particular mechanism in the early phase, your phase 1 and phase 2 of your clinical trial. You have the drug, and then you have the what is the molecular proof of mechanism going to do. It shows the proximity of biomarker to target. Is it like that is the target itself or upstream of, for example, your phospho-EGFR, your AKT, and your ERK. Then which tissue? Where exactly? Is it like your hair follicle, skin, or whatever? How much is it inhibition, and how long is the drug effective? Pharmacokinetics and your pharmacodynamics relationship. Then is it relationship with preclinical cytotoxicity and efficacy, and relationship with clinical toxicity and efficacy. This is also, here you have your mouse models. Then you develop your proof of concept. What are the functional consequences? Like, are they still growing? For example, you take the tumor and stain for your K67 KLT PET, then your metabolism, then angiogenesis, then DNA damage and apoptosis. You have your definitely certain endpoints here. What exactly? Where you have your molecular arguments and then your functional consequences. Now here we see what are the licensed anti-cancer drugs. This is very important, and some of the companion diagnostics to this particular drug. Imatinib for your then seek it, and then it's in the KIT mutations. Then this is a very important drug, trastuzumab for your head to for in the, and this is all the test you have to do for that is your IHC. So from the pathology it has to be done. Pertuzumab for your head 3, and then this is in the IHC it's done. Then here you have the molecular diagnostics coming up here. Then where you have your erlotinib, efitinib, debrafenib, vemurafenib. So this is for used for your EGFR, where your L0 efitinib. And how do we check for these mutations? Using the qPCR or your NGS panels and all that. Then you have your trematinib for MEP, and then your crizotinib for your ALK. So then again tamoxifen for ER positive. This is for the breast cancer. This is all different targets are there. Now are we using each drug only for one cancer? No. This is where I will clearly slowly come across the different clinical trials or how they should be. It is like you have the BRAF mutations here. So for your and that they are there in your melanoma, or HER2 mutations which are there in the breast cancer. That is, you have a drug that can target an aberrant pathway. So now this aberrant pathway may not be specific only to that particular cancer. So you have them as a specific genetic. This particular drugs can target the pathways in all different cancers. Please make this point. This approach does not always lead to finding populations of patients with mutant oncogenes. You may have to end up screening people for their different mutant oncogenes every time. Several cancer boards, your national comprehensive cancer guidelines, all of them, they recommend to go for different mutation testing, for example for lung cancer. It is a mandatory regulatory approval to go for mutation and amplification status of the tumor or molecular profiling of the tumor before the therapy is started. Now, what are the different endpoints? You need to have intermediate endpoints of response along with your molecular markers. This is how the typical tumor baseline. Maybe you want to know that the tumor has reduced. Then you have the intermediate endpoints such as your PSA, then your CA125, then you use your markers for the mutational markers, then you use your PET and your MRIs. Then you check for the sampling of biomarkers to detect early progression, your CTC, cell-free DNA. And then you look for mutations and the gene copy number. And again this response, then progressive response disease. Now you can see the tumor size has increased. Yes, the tumor, it didn't really respond. Then you take the tumor biopsy and look for any novel mutations, gene copy number variations, gene expressions, and then you look for proteomics. So intermediate endpoints of response and reassessment of the disease during progression, these parameters are very important. And here we show that what are the tissues and what sometimes even have the blood also to evaluate this blood markers. So it is very important to identify validate your surrogate endpoints, such as your technologies what we have seen, such as your ctDNA, then your machine learnings. They can help to identify your surrogate endpoints and potentially expedite the time and your cost of. It's always easy to take a blood, as we have seen, rather than to take a biopsy bit, right? The science of clinical trials in is oncology has evolved to including your phase 1 dose finding trials and phase 2 studies to establish the efficacy in a single tumor type, through phase 3 trials to compare the standard of care with potential advances, with maybe compare your original drug with a different with another standard drug. And then, so when you are doing so much, what is it, what can you expect? This much of response, the patient is a response. It is like higher cure rates in locally advanced cancers. So this is a very very typical approach. You have the, this is basically a statistical approach to categorize your clinical trials, where you have the frequentistic approach and the Bayesian approach. What happens here? You fix your design, set analysis plan, monitor and oversee your data collection, then conduct interference based on point estimates, then look for coincidence intervals. Here, what does it happen? You have to have your simulations, team deliberations, and you take lot of preliminary data to design. You need to have lot of parametric prior probability distribution to decide on your designs, and then you collect data and use to update parameters based on the posterior. Then you have the conclusions here. What is an enrichment trial? Trail designs called enrichment designs or targeted designs. They are studies with patients populations with a single molecular marker for which a drug's effects can be expected in a specific tumor type. So very very important, single molecular marker. This is very much a specific. This is a complete targeted design. This design is selected on the notion that, you know, the molecular marker is an established marker which is strongly correlated with the efficacy of your investigation drug. Yes, so I'll tell you the example. It has been biologically demonstrated that cannot be drug efficacy cannot be expected in the marker negative case. For example, I have an HER2 negative. Yes, I can say that yes, this drug will not work. This is such a very fantastic and this is one of the very selection assumption for the enrichment design trial. And there should be a diagnostic tool for this molecular marker. This diagnostic tool should be like, suppose you take pre-treatment one biopsy, then after some time during the treatment you take a biopsy, maybe IHC, and then after treatment, then how is this particular marker stated is evolving. So a companion diagnostic you are supposed to have. So you have very importantly trastuzumab. This is a very very important molecular enrichment design. The enrichment design clinical trial, N9831 trial for HER2 positive breast cancer. So, and even for your TOGA trial on HER2 positive stomach cancer. This is very very, and then so you have identified a biomarker, HER2 positive or negative. You can do an IHC or your FISH. So if the molecular marker is not able to establish, if only my molecular marker is not good, the use of your marker stratified design may be considered. You in this, what will happen here in this design? You patients are assigned to marks to arms by molecular positivity or negativity, and you will randomly pick patients within this arm. What happens? A master protocol trial, it uses a common system for your patient selection, logistics, templates, and data management. Though histologic and your hematological specimens of your patients enrolled in master protocol trials are measured using your common basic system like your IHCs and your NGS, and then you collect the completely coherent molecular data. So patients can participate in sub-studies in which they meet eligible criteria based on their molecular marker data. Thus, enrolling in a master protocol trial, it will increase the chance of participation of people in a trial that is most suitable for a given patient. Even if there are no sub studies that a given patient can participate, they will be followed. For example, the patient is positive and we know that his mutation status is such that he will not be able to take the drug. They can be placed on waiting list until unless for the different other study is started. This is how it uses a common system for your patient selection. You have this is your analysis of your genomic information using NGS, marker 1 positive, 2 positive, negative 4, all positive. You have the marker 1 positive, sub-study type 1, sub-study type 2, and then a different so on, and you can have as many sub-study types based on the different particular drugs. You should need to have a, yeah, that's very very important. You should need to have lead drugs. Histological and hematological specimens are enrolled in the master trials are also measured and analyzed using a common basic system, as I mentioned. So this is like the natural history data from a waiting list can be used as control. This may be in evaluating the efficacy of the drug in this particular single arm studies. Suppose you have can we use this as, you need to have a different specific control. You have common protocols that assess the combination of several molecular markers and their targeted therapies by means of multiple sub-studies, and they are required for single or multiple tumor types. These protocols are master protocols and are driving attention for your next generation clinical trial designs. Trial purpose master protocols, they can both be exploratory or confirmatory. There are exploratory are mostly single arm study sub studies, and confirmatory master protocols are multiple randomized sub studies. For either trial type, the design and statistical considerations are common standards for all sub studies. Different form of traditional, one indication at a time. That is a this is completely different. You need to look only at one indication at one particular time in a clinical setting. We have that only test one drug or one histological parameter in a clinical trial. Master protocols, they evaluate multiple drugs, multiple histologies, or multiple molecular subtypes in parallel. For example, you have an nanostring, you have different 360 panel. You can have a different from using many other tools. You can have TCGA data sets and you can compare, and you can clearly say that if this particular molecular subtypes, can they have different drugs, or you can clearly check how is their progression in response to this particular drug. Such multiple drugs, they allow logistic efficacy. You have centralized screening, then you can have biomarker profiling, and then this can lead to a much accelerated clinical drug development process. That is why we are calling this next generation clinical trial design. So there are you have three types of your master protocol types, such as your basket types, umbrella, and platform, based on the characteristics of the study. Example, the population, disease, histological types, molecular markers. Based on that, what does basket type do? It evaluates one targeted therapy on multiple diseases or multiple disease subtype. Umbrella trial type, it evaluates multiple targeted therapies for at least one disease. Then platform, it evaluates several targeted therapies for one diseases continuously and further accept additions or exclusions of the new therapies during the trial. This is a very very important definitions which we all have to keep in mind. What happens in basket trials? So it evaluates one targeted therapy on multiple diseases subtypes. You have tumor type A, tumor type B, or then tumor type X, Y, Z. Different either the same tumor, different types. You have a marker A plus a targeted drug A. Then you have the marker N targeted drug. Then you are stratifying this. This is one more of lalotrectinib was approved for TRK fusion cancer, positive cancers in adult and children using this particular basket trials. They have the therapeutic this you exemplify the therapeutic effects of molecularly targeted agents for several tumor types that may have a common single marker or a genetic mutation. So in this scenario what will happen, the group tumor types form a basket and studies are conducted by tumor groups within it. Basket trials are often conducted as a single arm phase 2 trials with the purpose of evaluating your proof of concept in a early stage of development. Generally, the number of participants in the individual sub studies are usually between 20 and 15, and hypotheses that can demonstrate statistical significance are made only when there is a major therapeutic efficacy. So it is like a signal finding trial. Signal, you find, yes, there could be a lead if I use this particular drug. Two-stage or a multi-stage device for the may be used. As such, basket trials are characterized by the comprehensive execution of single arm trials with a small number of patients, which enable efficient enrollment for rare cancers or for rare fractions. Basket trials have the assumption that they allow a fairly accurate prediction of whether a tumor with particular molecular characteristics will respond to that particular targeted therapy, irrespective of the histological type of the tumor. Yes, can you have this? Is usually you have the molecular markers as well as your histological markers. You can have this basket trials designed to test one investigated drug or a drug combination in multiple cancers or cancer subtypes with a common biomarker linked to the therapeutic target of the drug. As I mentioned that your larotrectinib was approved for your TRK fusion positive cancers in adult and children. They are accessible for patients with rare tumors. This is when so you may have a challenge that that the molecular variants may not be the only driver of tumor response, and unpredicted heterogeneous. We are not talking, as I always mention, these tumors are always heterogeneous. So, for example, you are giving only a target drug to one particular marker. They may not be very much effective. That is like, so for example, you have your vemurafenib, which works in BRAF mutated melanoma, but it doesn't work in your BRAF mutated colorectal cancer. It was proved. Either you use this particular trials in pooled analysis across tumor types or independent test for this is your pooled analysis for many tumors, or you use your different independent tumor. What happens in the pooled analysis types? It ignores you will not, you are totally ignoring your tumor heterogeneity effect, and then you may have, like, suppose one individual may not hasn't responded, you may have to say that it is going to be a failed study for all individuals in the basket trials. It is, you have to be very careful to tackle this challenge. Many basket designs have emerged to tackle this particular challenge. One more very interesting type design for your umbrella trials, that is your apart from your basket trials. Isn't it very fundamental, very nice picture? They evaluate multiple targeted therapies for one disease or several disease. For example, they are expected to respond to an investigational drug. In oncology, sub-studies are conducted to evaluate targeted therapies that correspond to different molecular markers or genetic mutations within a particular tumor type. In that case, the tumor type is your umbrella under which sub-studies of each molecular marker are operated here. You have the different molecular markers. Then sub-studies, again you may have even a single arm, or which can be phase 2 or phase 2 or 3 trials, or and they will be compared with your standard therapies or your standard placebo. And umbrella types have a common system that unifies molecular profiles of patients' serum to your umbrella. So while basket trials are generally arm specific sub studies that are exploratory in nature, umbrella trials are often single arm or randomized sub studies that are confirmatory. So you remember, we have exploratory master trials and then you have the confirmatory master trials. Your extrapolatory are in the basket trials, and the confirmatory are coming to your master trials here. So, but the umbrella trials are normally usually large scale and long term master protocol trials. But when the standard therapy comes during the therapy, the clinical significance of comparing to a control group of patients undergoing standard therapy is lost because it may take a little longer. So, umbrella trials are also termed as molecular allocation studies. So you have the control here. What is your control is like, as I mentioned, your different standard treatment or your placebo or anything. Marker A, targeted drug, and then this is a very important master protocol type, which is your umbrella types. You have your basket designs. For example, you have the VE basket, which has in the early phase 2 BRAF mutation. It is like testing for the efficacy of your vemurafenib in patients with BRAF V600E mutation. It was usually in squamous cell lung carcinoma they have tested, and then colorectal cancer, and then Edmund-Chester disease or Langerhans cell histiocytosis. So here they found it is a 42% ORR, but whereas this is 0% in colorectal cancer. Then again, you have your LOXO TRK 14001 Scout Navigate phase 1 trials for NTRK fusion to test the efficacy and fusion in NTRK fusions for patients with NTRK fusions. Then you have your ALCA START TRK1 and START TRK2 phase 1 and 2. This is your basket trials. Then coming to your umbrella trials, which are well validated, you have your lung map, then then you have the alchemist, which we will see in detail later. Now we will talk about the different clinical trials for which are currently used in oncology. Coming quickly again back to our different biomarker, companion biomarker identification, then you have the assay development, then look for the clinical assay application in drug discovery, then assay validation, then clinical validate of your particular drug, then get the regulatory approvals, and then how do you incorporate in your clinical practice. These are your different anti-cancer drugs such as your cetuximab and your panitumumab for your CRC, where and then the markers are the RAS mutations. You have the PARP inhibitors for ovarian cancer. Then for the melanoma, you have the dabrafenib, vemurafenib, and trematinib for the BRAF mutation. Then you have the eltornib, imatinib, pembrolizumab. We have studied all this, right, for your immunotherapy. Then trastuzumab is for the very important, one of the very important trials which we have studied, enrichment trials, and nivolumab for your melanoma for the BRAF mutation. What are the samples you take? Either you take your liquid biopsies, that is your blood for circulating tumor cells, circulating tumor DNA, then you take the exosomes, microRNAs, or you take even directly the tumor biopsies. You have the CT guided biopsy where the tumor tissue is excised and the IHC is done, and you embed this tissues in the FFPE. We have gone through this thoroughly. Their basket trials are basically histologically agnostic studies that typically focus on variety of tumors. Umbrella trials use multiple drugs that target multiple mutations in a range of tumor types and in the context of a single study. Very important. They may be randomized or non-randomized. So, N of 1 clinical experiences in which a patient with a tumor type that is unlikely to undergo an objectively defined response demonstrates local clinical very robust clinical benefit from therapy. And if you can characterize the tumors from such patients and can define unique molecular signatures that are really very well responding to this particular therapies, you can really come up with the predictive signatures power after the clinical trials for a defined patient population. So this is how basket trial. This is how a very good basket trials. You can you have the lung cancer where you have the basket trials. Then you have the umbrella trials and then the exceptional responder trials. What happens here? You have single drug targeting a single mutation, variety of tumors carrying a genetic aberration X. This is a single drug. You have drug A, drug A. This is a how so, but these tumors are different. You have the lung, the colon, and the liver. But whereas in the umbrella, what is happening here? You have the same tumor type and again even a variety tumor type. And here also in the same tumor type, you have a different. See here, you see it's only a single mutation, only a single molecular difference. But here you have a difference. You have XYZ here in the lung, XYZ in the lung, and even in the liver and in the colon. So you either have a randomized or a non-randomized. These are rules-based treatment. You have assignment of per patient based on the review of the individual profile data. You have different drugs. For example, X, the drug A is coming up to your X mutation. Then you have drug B to Y and C and D. Whereas your exceptional responder trials, any cancer type and drug where a patient had unusually robust clinical benefit. This is the very fantastic part. You have the drug exceptional response, then this it's coming down. You can see the size of the tumor coming down. Then you will find the molecular signature X in patients with exceptional response. You will find molecular signature Z in this particular patients with exceptional response. Then conduct clinical trials in the molecularly defined patient subpopulations. You are totally stratifying the patients based on the tumor molecular or exceptional response or one-of-one clinical experience in which any patient with a tumor type that is unlikely to undergo an objectively defined response demonstrates robust clinical benefit from therapy. This it really very it looks very, it sounds very fantastic, but it is like characterization of these tumors from such patients. You can identify unique molecular signatures that can be tested for their predictive power in subsequent clinical trials for defined patient populations. What is the level of what do you mean by this level of evidence for prioritization of actionable molecular? While really talk expand this NCI match trial, what it is? You have a gene variant credential. In this one, you identified drug and then a gene variant. Then molecular characterization is an eligible criteria for an ongoing clinical trial. Utilizing the drug or the abnormality has been identified in the N of 1 clinical response. Then you have the clinical response in the level 3 where models with variant respond while models without. So for example, if there is a mutation, yes, this mutants one will respond to that particular drug while the people without mutation or they do not. Then you have a gain of function of mutation demonstrated in a preclinical model. And loss of, for example, your tumor suppressor gene or which is whose function is lost. 1CI match stands for National Cancer Institute Molecular Analysis for Therapy of Choice. This is a very fantastic trial. It's an umbrella study to examine the hypothesis that treating adult solid tumors and lymphomas with molecular targeted therapies independent of disease histology can be effective. I mean, this is a very fantastic that usually they find the response rate. Over 6,000 patients have been accrued at over 1,000 clinical trial sites. Initially, all this 6,000 patients in this particular trial underwent fresh tumor biopsies for next generation sequence by a centralized laboratory network, very important. And then 30 drugs from a wide range of pharmaceutical firms, they permitted accrual of the patients with low prevalence mutations to a series of phase 2 investigations. Very interesting study. So it is composed of 24 sub studies that evaluate the efficacy of at least 17 of your targeted therapies for solid tumors and lymphomas that were treated for at least with one regimen. So your response rate is the primary endpoint. You have the genetic, as I've said, you have identified the actionable mutation, then you have the study agent. Is it stable? Stable disease, complete or partial response, and then continue on study agent. Suppose until progression, then your progressive disease, and then if it is disease is progressing, check for additional mutations. So maybe this particular drug was this particular patient was wild type for that mutation. Now did he develop any mutations, that is why he is getting resistance to that particular drug? Yes, again, if you follow the actionable mutation, is there a chance further to go down? No additional mutations were detected, but still there is it is progressive disease, he's still progressing, then the patient can be removed off this. This is a typical very well document, very well algorithm for NCI match, NCI molecular analysis for therapeutic choice, molecular analysis for therapeutic choice trial. This is very important algorithm where and it is one more very important algorithm is your alchemist, not a novel alchemist, but adjuvant lung cancer enrichment marker identification and sequencing trial. Very interesting trial here, where ALK and EGFR positive patients are enrolled in the phase 3 randomized sub study, which comparing your so you see in recollect in my first slide where I showed about the inhibitors, or a randomized comparing erlotinib. For ALK positive patients, they use this drug to placebos. The primary endpoint is like your overall survival, and an interim analysis is planned. The patients who were both ALK and EGFR negative, PDL expression 1 is measured, as they are considered for enrollment in a rand. This, if you know, we have one immunotherapy target, if you recollect, that comprised nivolumab administration group to an observation group. The primary endpoint of each trial is overall survival, and an interim analysis is planned. So this is one more example. This is their alchemist trial. Here you have your very much very interesting with resectable lung adenocarcinoma, and in this trial they have screened tumors for their EGFR ALK mutation. This is your workflow. So the clinical TNM status was taken, pre-operation option, then your after the operation also the TNM status, then you stage the tumors or the histopathologically, and then look for the age, then local test, look for the qPCR for your EGFR and ALK, and then the patients have to go through the epidemiological questionnaire, and then all this particular details I've taken. If it is like if EGFR or ALK positive, assessment for adjuvant treatment trials, yes, went about. EGFR or ALK negative, willingness to be followed for recurrence for 5 years. They should be in part of, yes, the follow up has to be there. This is the workflow, how it goes. This typically explains how a drug discovery trials go on. FFP block, then you have the questionnaire according to their epidemiology. If the patient is not resectable, or like that the surgery is not going, and the TNM does not fit the eligibility criteria, the patient is off the study. Then again after operation FFP, then go for the genotype, slides for genotyping, scrolls for genomics. Then you have the screening component, PC testing for EGFR mutation, then ALK red mass rearrangements. Patients whose tumors cannot be evaluated by the central lab, in the central lab, if they were in, suppose for example, if the quality of the FFP is not good, they were moved off the study. If EGFR positive, what is the treatment trial? They have given this particular treatment numbers, then they are given for this particular enrolled on adjuvant treatments. If EGFR negative and ALK negative, You know what did they do? They went on for the research companies where the blood, it's not only your FFP, they even blood sent directly to NCI for them for DNA germline analysis on uh, patients associated with the genetic research. Then they have sent this particular tissues to different places for genomic research, deep sequencing, and exon sequencing, and then evaluation of the epidemiologic correlation. Observational study with to occurrence for how was their study for 5 years? How did the patient keep at recurrence? Patients should undergo biopsy to confirm recurrence if deemed clinically appropriate by treating clinician with some tissue donated for research. Without these mutations, they for patients without these mutations, they will be enrolled in a registry study and have their tumors undergo whole exome sequence. If they relapse, every attempt will be made to resequence their tumor DNA at that time to determine the natural genomic history of this particular. This is a very wonderful algorithm, alchemist trial. For example, how and it's a very 6,000. This is and that too for lung cancer. Difficult to get your biopsy beds. That this was which was very well taken forward in precision oncology. So these are the different trials we have. You have the B2225, which is a phase 2 multi-center unblinded uncontrolled trial, and they have tested on imatinib on 40 carcinomas. 186 patients were enrolled, and the response rate is the endpoint. So for many of these trials, your BRAF V360 is also a multi-center unblinded uncontrolled trial. Vemurafenib and cetuximab for on multiple class carcinomas the disease was tested. Then you have 122 patients, the ACAC, which used crizotinib on gastrointestinal, breast cancer, ovarian, thyroid. Almetros, Ron Fusion, 488 patients were enrolled. NCI match, which we have seen for EGFR MET, AL cross. So for your lymphomas, melanomas, and then you have your I spy 2, Shiva, and the NCI 2. This one is for your different erlotinib. This is for ALK mutations, and then lapatinib, trastuzumab, sorafenib, imatinib, desertinib, vemurafenib, all these drugs, tamoxifen, and then you have the different targets. Then this is for all advanced tumors which are resistant to standard treatment. What is the endpoint here? They would assess the patients, 195 patients who were enrolled, and it should be a progression free survival. This is how they have employed different clinical trials. Similarly, you have your alchemist, which we have just discovered, and then we have the lung map, seamless phase 2-3 randomized trial for, and then you have 170 patients in the lung in the phase 2, 400 patients in the phase 3. Then you had the focus 4 phase 2 phase 3 designed multi-center unblinded randomized trial. You had drug targets such as your BRAF inhibitors, and then you have the battle 1 trial, phase 2 single center adaptive randomized trial. Many of this particular drugs were used, and you are targeting your EGFR, KRAS, BRAF, VEGF, RXRs, and cyclin D1. Now, after talking about so much of precision oncology in drug design, what are the important drugs, what are the clinical trials, what are the different clinical trials, and then now shall we just come to a very team effort. It's not like it's like now we now say that cancer doesn't fit, one size doesn't fit all. It's so now you need to have each patient's treatment strategy has to be tailored to fit at home. Then how many people would you need? This is not like a single person effort. You need to have a multidisciplinary team effort. You have the core team and the non-core team, where the core team consists of your immediate surgeon, pathologist, then your oncologist, and the non-core is your genetist, then your physicians, nuclear medicine, palliative care. This and here your importance of your molecular biology or your molecular biologist or your molecular scientist very well important here. So it's not like that. How do you take decisions? How do you take decisions that you're going to have this particular patient given this particular treatment? No, it is not an individual decision. It is a this particular patient is responsibility of that particular organization. Then you need to have a very well placed tumor boards to take decisions. But now we are coming to another term called as a molecular tumor board. Very important. If it is like a molecular, you had a regularly a tumor board only where you had a pathologist, radiologist, surgeons, and then your medical oncologist. They will give a diagnosis. You have your PET CT, then you have the pathology. You subcharacterize based on the histology, and then go for treatment, surgery, chemotherapy, radiotherapy, targeted therapy. That's all this year. But whereas a molecular tumor board, so just this advancement, the whole thing has taken a drift with your evolution of your NGS, next generation sequencing here, where you are giving so much information about the tumor. It is also very much a challenge for the clinicians to take the right decision to give the right treatment. You have the diagnosis, you have a bioinformatic platform, you have a genomic platform, and then you have the database. Can I compare? Is it the first mutations which I have seen in this cohort of patients, or are they there? Yes, there's so much of data available in your TCGA data sets. And then you have the treatment. Apart from your regular treatment, you even have your targeted therapy or your clinical trials. So this is something which is very, very important part. To have a particular clinical trial, you need to have your molecular tumor board. Due to the impact of molecular biology as a tool to support different therapeutic decisions, there was a need to add to the standard. So, molecular tumor board comprises of series of specialists focused on molecular biologists, different scientists and counselors. What does this impact? How does it impact on the clinical management of patients with cancer? You have here, it aims to improve your patient management and outcome. Yes, definitely you have much of information about the tumor molecular profile. Yes, you can give a better outcome, better result. And then you have a local multidisciplinary oncology group which will request your MTB consultation. That is your molecular tumor board group. And then molecular tumor board will have a discussion, their meeting about all the profile, and then you will have a central regional, so it's not that only one testing for this tumor is done, it's even done at different levels. And then you have the biomarkers report and MTB interpretation. This data interpretation is very, very important. You should be able to say yes, how this particular gene expression, for example, you have your BC316, your nanostring, how is this gene expression related to the biomarkers report and MTB clinical interpretation. Then database consultation and patients personalized treatment recommendations according to your local emoji. Standard therapy and off-label clinical trials. Yes, is it improving the patient outcome? You have to adhere to MTB is molecular tumor board's therapeutic intent. It is very, very important for the best national and international guidelines. Is one of this is a very important factor for assessing your quality of your molecular tumor board. It is very much higher adherence to current guidelines has been observed for both staging and treatment. One more very, very important part is your MTB recommendations is your documentation. MTB recommendations should be clearly written on ad hoc design reports documenting parameters apart from the patient information such as the age, sex, smoking status and all that, which is regular by part of the by virtue of all your electronic data, medical records will carry all that. So you should this particular documents should have your driver mutations, copy number, structural variations including your fusion genes. Then you should be able to say druggable molecular alterations, microsatellite instability, tumor mutations, very, very, very important. TMB. How are these alterations indicating drug resistance? Then MTB conclusions, recommendations, and then potentially available They should be able to tell this particular clinical, not necessarily all drugs in clinical trials, but even some particular drugs which are in the market, or also this MTB has to be able to, molecular tumor board has to recommend. Then you have your a very important data. You have your pathological data, you have the imaging data, then what is the available treatment, as I said, multidisciplinary tumor dose, all the patient characteristics. Isn't it very fantastic image, this one, where you have your molecular information and then you have your molecular tumor board. It's not necessary only the oncologist uh, which are only taking part in this decision. Sometimes all oncologists may not be well informed with exactly the different molecular profiling techniques and the terminologies. Yes, that is the reason why genetics and the molecular scientists are part of your molecular tumor boards. They should always be patient identification, reporting style, interpretation. They MTBs can facilitate clinical trial involvement and off-label treatments through an online database consultations. They have to be very helpful in selecting the most appropriate biological samples. In current clinical practice, your liquid biopsies is used for the identification of your driver mutations and circular ctDNA, with the first clinical applications in your lung cancers they have used this. Now, your compliance to international guidelines. This is very, very, very important. You have the several international guidelines given by your NCC and National Comprehensive Cancer Network guideline recommendations. Only if they recommend only any biomarker or your companion diagnostic test, they can be taken. And adherence to NCCN was observed when they have studied, when there's a very recent report from Cell on 2022, and they have taken how this MTB is molecular tumor boards are functioning. Are they really adhering to the NCCN and guidelines? Yes, 98% assessed in the only are only 80% evaluated by single specialist received a therapeutic indication by NCCN guidelines. So only very few deviations, especially for based on patient's age and comorbidities only. When the MTP hasn't indicated, molecular tumor board hasn't indicated, only there are deviations. For example, they have obesity or a diabetes comorbidities. Then maybe your regular clinical trials or cannot be, these patients they have to be excluded. It is a very, this particular group has found an adherence rate to 100%. There is but clinician adherence to the treatment plan recommended by the, what happens, an MTB molecular tumor board recommends a treatment plan. Is the clinician, can he adhere it? But it may not be further very, very, very complacent, because the patient may not be for cooperative, or maybe they could be more further diagnostics and investigations or medical conditions should be needed. Which tumor types should be discussed in your molecular tumor report? It should be all tumor types are eligible. So many rare malignancies mostly represent the majority of the molecular tumor boards. You have the breast, brain, gynecological, lung and colorectal cancers. These are all falling in the molecular tumor. And very, very important parameter is your next generation sequence. It should be very much considered as an integrative part of your histological diagnostics for malignancies which lack your standard therapeutic options. What molecular analysis could be performed? What should you, apart from NGS, you need to have your whole exome sequencing, transcriptome, then you have your array comparative genomic hybridization, whole genomic sequencing, Sanger sequencing, and mRNA sequencing. All this were used, this particular tests were used for discussion in your molecular tumor boards. The most appropriate analysis with the aim of finding actionable alteration is target NCS sequence of the most important cancer associate, ideally analysis for clinically relevant, example gene fusions for your kinase fusions. And so this is like molecular test recommended by your treating oncologist, they will recommend, and then case is referred to to the molecular tumor board by your treating oncologist or pathologist. So discussion of the case in your panels, and then one of the recommendations the tumor board means continuation of the ongoing existing therapy only, or in case the tumor board feels because of the tumor mutational profile the patient may not respond, can you start a new therapy? Then should it be do you need to have a new molecular diagnostic test, and then enrollment in a clinical trial, and then all these particular recommendations have to be entered in the clinical in your electronic medical records for access for treatmenting oncologists. How do you should molecular findings be classified for drug selection? Yes, back to our old questions, what we have clearly seen before. Based, yes, your molecular findings have to be classified for. How should you classify it? You either take your blood tissue sample of histologically confirmed solid or hematological, and then go for sequencing and molecular uh, profiling through particles through several NGS platforms, Illumina platforms, then put it in the board for different where your different uh, members from your other research, palliative care, and hospital ethics committee are present. Then recommend a clinical recommendations such as your current matched clinical trials or summary of molecular findings or matched therapies with clinical benefit. And then this is like how your there is an increase in the rise of your targeted therapy, or you are more talking about precision therapy or precision targeted therapy. There are this is a different parameters of classifying molecular alterations based on the potential clinical utility. You need to have very good progression free survival. You should have important overall survival for the drug to be very successful in precision oncology. You are having very good array of drugs, and you have identified the companion diagnostic markers for these drugs, and you are looking at how this endpoints when you are giving to this in the during the clinical trials for the patients. Now, how do you really it may look very, we have now gone through the complete details of how the drug discovery process, then we went about the different trials process, what exactly are the master protocols, then basket trials, umbrella trials, then we said that a single tumor type for basket trials, and many tumor types and many market markers for umbrella trials. Now after seeing, then we came how is it adapted in a hospital scenario by selecting use of your molecular tumor boards, your MTBs here. How does it is it all may seem, but of course there is a lot of limitations when it comes to your, yes, many key hospitals, many important institutes in India have all this molecular tumor boards. Yes, but is it there is a lot of limitations. It is pragmatic, evidence based, contest adapted tools, so based on your molecular biomarkers. So methods to detect they have been to detect your mutational signals have been established in your research settings, but of course they have been very well adapted in the clinical research also, but still a lot of challenges are coming in play. For example, for your analysis, so you complete an NGS profiling and one the analysis. So how do you make a complete only if an analysis is done by even a small lab technician also, can you have a very good authenticated result? Yes. Tissue which is very important. Specimens for clinical molecular testing contain low tumor purity because of the proportion of neoplastic cell nuclei towards all cell nuclei, because the proportion is very large. So it may not, you may not be the during a biopsy you may not be excising only the tumor tissue, sometimes even the normal tissue comes. Then how do you does this impact? Yes, then your low tumor purity, it impacts the ability to detect somatic sequence alterations near the limit of your detection and severely limits the ability to detect your copy number changes in a panel of sequencing assays. So in tissue specimens with these limitations, you are having the accuracy in detecting will depend, so it's reduced. So you have one more very important part is here is sequencing of age formalin based parafilm FFP blocks. So as the archiving goes more, so you to extract the DNA and the RNA and then have a very good yield of that particular DNA RNA, and so you are not able to get a very good specimen sometimes. Then definitely if I want to check for the original mutations every time, maybe after 2 years 3 years, it is a real challenge. So how do you maintain the so and then and the limited availability of page sequencing of non-neoplastic samples to the filter germline variants. This is also pairing of samples every time. Suppose for a lung, I can't take a tumor of bit of the lung and a normal, right? It's a very very precious organ for the human being. You cannot really have a normal or a non-plastic neoplastic samples. That is when we use a database to filter your germline variants. But how can it be same from for all individuals? This is an additional very much challenge in your clinical practice. Then you have your ethical challenges. How do you to maintain and improve the trust of the patients, then your authenticity and of your study, and then you have to look into many clinicians, researchers and industrials in academic medical fields. This is all this is a very much a limitation in your clinical practice, and definitely a lot has been explored in the last 25 years after the human genome project was is over. A lot in precision oncology has happened, and definitely the many studies are on the way, many clinical trials are on the way to improve the clinical outcomes for patients suffering with many different rare cancers as well, and also to give treatments which are only specific for that particular cancer group. Thank you.